Not too loud though. I get pretty excited, so might be doing yourself a disservice here telling me to put myself on a louder volume. Um, well, I am very happy to be here today to present to you some of my research. It was said that these two presentations were pretty different, but I actually think that they were different enough to compare and contrast in an amazing way because that was an urban environment, and I'm going to actually take us into more kind of pristine environments where trees do and are able to reach ages of 200, 300 years of age. So, that being said, I am a research scientist at the University of Washington over here in Seattle, so the Pacific Northwest coast in the States. I have been here my entire academic career. I got my undergraduate in restoration ecology and environmental horticulture. I got my master's in soil ecology, and then I got my PhD in fungal ecology. And now I am working on biotechnologies, bacterial fungal interactions, and I also am still working in these forests. So I'm very excited to kind of give you a crash course of a lot of the research we've done out there. The way this presentation will be set up is I'm going to give a little introduction to fungal organisms. I just think it lays a good foundation for the presentation because I'll be using a lot of fungal centric definitions and words. And then we will kind of lay out a bunch of different projects that we've done in this area. I've been doing research in these old growth ecosystems for the last 15 years, and I hope that I get to do research there forever. It's really amazing. So the topic of my discussion today is don't forget to look up and why canopy soils are so important to old growth ecosystem function. And this is just a list of amazing collaborators and people who have made this project possible over time. The list extends, but then that would just be a giant slide of names. So they know who they are. They know that they're integral to this project. So, before we hop into the research, I just want to emphasize how important fungal organisms are. I don't really necessarily look at myself as a tree scientist. I am more of a soil scientist that looks at how different organisms interact with each other to fulfill ecosystem processes. So, whether that's how fungal organisms interact with trees, other types of plants, or other organisms in their environment, and also the collective circle of how all of these things interact together. So one of the important kind of baselines I like to start out with is that evidence shows that when plants evolved on the terrestrial landscape, so we're talking in the scale of millions and millions of years ago, there is fossil evidence that shows that fungal associations with plant roots, or what earlier evolutionary rooting traits would kind of look like, but it shows that these fungal associations were vital to the establishment of plants on land. And this association, actually, I'm just going to change my pointer to the laser here. So this association of fungi and plant tissues is very consistent in fossil evidence. So when they look back at the fossils, there's always fungal filaments with those early lineages of plants. But what they also saw is that through this evolutionary timeline, so we move out of kind of the mosses, the bryophytes, we go into the ferns, we're starting to get into seeded plants, then flowering plants. As we go that direction, these fungal associates diverged many times, but what stays consistent is that about 85 to 90% of plants globally form mutualistic relationships with what we call mycorrhizal fungi, which are not the only plant mutualists. We'll talk about that in a second. But basically, this plant fungal association has stayed consistent over these millions and millions of years, and they are very important to how plants survive and also how ecosystems function in general. So to kind of just break it down into the groups of fungi that are in ecosystems that are really important, this is very much a basic kind of outline. I could give lectures on all of these groups for, an entire semester. But we basically have decomposer fungi. So there's, for example, white rot fungal organisms. These are powerhouses. So decomposers are so important in ecosystem function because they break down organic matter. Specifically, white rot fungi can break down lignin. So lignin is this extremely complex compound. And I always like to shine light on fungal organisms because they are the only organism who have evolved over time to efficiently break down lignin. So you can kind of thank fungal organisms because if it wasn't for them, what would our built environment even look like? You don't want to build things on organic matter. If fungi weren't breaking down organic matter, it would not break down as quick. Some things may never break down fully. We don't know because fungal organisms have always been here. So they are really important in breaking down these large products and returning these nutrients to the ecosystem. There's also brown rot fungi. They can break down cellulose, so some of the byproducts of lignin. There's other decomposer fungi. And what this is really just painting a picture of is how important these diverse groups of organisms are in ecosystem function, not only in forests, but for example, if we go into an urban environment and there's not a lot of soils or the soils are really unhealthy, we might not have as many beneficial interactions that are able to drive the soil health in a beneficial way. So decomposers are important. As we look into this society, I think that pathogens might be brought up more often. I'm not sure, but in arboriculture and arbory, I think that pathogens get a lot of light shed on them because they're always present in ecosystems and they are typically what cause the diseases. So, for example, root rots, heart rots, blights, rusts, those are very common in different types of ecosystems, whether it's a urban park, an old growth forest that had a disturbance, a prairie ecosystem, cropland. And so when we start looking at pathogens, fungal pathogens to be specific, we want to kind of step back and understand, as Levon was, kind of laying out with this tree health, that the health of an ecosystem is a partial determinant of this pathogen establishment. So, for example, fungal pathogens would be more prone to attack a street tree than they would to attack a tree in a healthy, pristine forest. That doesn't mean that when you go into an old growth forest, there's not pathogens. Pathogens are actually a part of ecosystem processes. You will not ever see a healthy old growth forest without some sort of pathogen interaction. So in these healthier old growth ecosystems, pathogens really do influence forest succession. And that's when you'll see a lot of these pathogens kind of acting as decomposers after they're a pathogen. So in old growth ecosystems, a lot of the times trees will grow really old. They will have a pathogen, but they'll be in a healthy ecosystem. Once that tree falls over, for example, from windfall, the pathogen can sometimes just act as a decomposer and then return it to the forest ecosystem. So pathogens in the sense of fungi are, kind of the bad guys. But when you go into the ecosystems that I've conducted research in, they really do influence the trajectory of those forests, and not necessarily in a bad way. So now that we've kind of laid the ground for the importance of decomposers and the importance of pathogens, whether or not it's an importance in the sense of mass die-off or survivability, we can turn to my favorite group of fungal organisms, which are fungal mutualists. And I want to lay kind of just a point down that I'm talking about these organisms as groups, but the diversity within these groups is vast. So I'm not just talking about a couple of species, I'm talking about thousands of species within all of these groups. And based on the conditions of the ecosystem will impact how they interact with that ecosystem. So the same thing stays true when we move into the mutualistic fungi. And we will first mention mycorrhiza, which is that fungal group that associates with about 85% of plants. So mycorrhiza, what it does is it establishes within plant roots, like tiny plant roots. So think of the size of like a pencil tip, or a piece of, lead refill for a pencil. These roots are tiny. So the fungal organisms, what they do is they establish structures in the roots. Depending on the type of mycorrhiza you have will reflect what kind of structures it has evolved to establish and also what kind of plant host it has evolved to establish with. So for example, about 80% of plants globally associate with arbuscular mycorrhiza. That includes trees, it includes shrubs, grasses, forbs, a huge diversity of plants. Ectomycorrhiza, on the other hand, only associates with about 5 to 8% of plants globally, but it associates with a lot of tall woody trees. So in areas like, for example, the Pacific Northwest, or when you get into other old growth forests around the world, you will have, I should say, especially in the temperate climates, you will have a lot of ectomycorrhiza. And ectomycorrhiza are a type of fungi that can fruit mushrooms. So This type of mycorrhiza that associates with about 80% of plants globally never fruits a mushroom above ground. Its reproductive structure is a spore, it stays hidden, it's very cryptic. And we never see it, but it is integral to plant survivability and has been since plants established on land. So I just like to reflect on the things that may be really important in ecosystems are often the things we may not ever see. And then we can get into the other groups and break it down. Again, I could give a lecture on all of these, but just the overall takeaway that there's different types of mycorrhiza. Depending on the plant will depend on the type of mycorrhiza. There's also some plants that can associate with both ectomycorrhiza and arbuscular mycorrhiza. It gets really complicated, which I love. So what the mycorrhiza does when it attaches to the root is it extends a hyphal network out. So for example, hyphae right here. And that can actually branch out really far and it uptakes nutrients for the plants in exchange for plant sugars. So it is this trade-off where the plant is like, here, have some carbon. And the mycorrhiza is like, thanks, I need that. It's the only way I can survive. Here is some, for example, phosphorus or nitrogen. And if the ecosystem is really unhealthy or something is disturbed, it can really impact these interactions. So then we talked about mycorrhiza, but what has been researched a little more often and what I'm particularly interested in as well is other types of plant growth promoting fungal organisms. So for example, there's a lot of root endophytes. So technically mycorrhiza go inside roots, but they are a certain group of mutualism. There's other root endophytes that won't necessarily be categorized as mycorrhiza. For example, these dark septate endophytes that we kind of see wrapping around the roots here. That can, for example, increase resistance for a plant when it's like in drought stress or if there's heavy metal pollution. Mycorrhiza can do that too, but we're just talking about kind of these two different groups of mutualists, again, stacking upon this complexity. There's, for example, leaf endophytes, which can produce the mycotoxins. It can reduce certain types of herbivory. There's rhizospheric fungi, so not necessarily fungal organisms that go inside the roots, but fungal organisms that have been categorized to hang out right around the roots. So some of those enzymatic interactions may be benefiting the root, even though they're not directly attached to it. It can get really complicated, and what's really amazing to me about this is research on this is really just starting to take off. Fungal organisms, mushrooms in particular, are very trendy right now. So I think that gives fungal organisms a moment to really shine when they've been buried underground or, for example, up in the trees, to really understand how integral they are to the survivability of our ecosystems. So now that we kind of have that overall backbone of fungal organisms, we can kind of think about how they provide, in a sense, of ecosystem functioning and ecosystem services. So I kind of look at this center line and expand it out to think about what are we missing when we don't consider certain parts of ecosystems. For example, if we don't think about what is above ground in certain forests, are we missing gaps in, for example, nutrient cycles, primary productivity, of course, biodiversity, and those are some of the things I'm going to get into. So the two sites that I have for this research are over here on the coast of Washington State, so the Pacific Northwest coast. I apologize. I realize I should have zoomed out on this map a little bit more. It's usually national or to Canada sometimes. So, you know, that this is the Canadian border right here, just to give you an idea. Seattle is right here. This is the Olympic Peninsula. If you are ever over in Washington and you have the opportunity to go explore these forests, I wholeheartedly would recommend that. They are not like any other areas I've seen in the world, and I've been to quite a few forests. So I have two plots. They are lowland riparian old growth ecosystems. One is in the Hoh rainforest, which is likely the rainforest you would have heard of if you have heard about our temperate rainforests. And then kind of the secret tucked away one, the Queets. So you can see in spatial location, they're pretty linear in where they fall in the sense of the watersheds. Both of these sites have similar species characteristics. So old growth maples, old growth Sitka spruce, big leaf maples to be specific. And then a lot of the understories, for example, sword fern, huckleberry, salmonberry. So a very well-structured, kind of, as I think, like locked in time forest ecosystem. These trees, the big leaf maples, reach over 200 years old. The sickest spruce in this area we reach well over 350 years old. A lot of them have been tagged to 600 years old. The oldest one out there that they know of right now, I think, is just under 1,000 years old. So the trees live pretty long around here. And so what is pretty focused on the climate in these areas and what allows canopy soils to form, which I'll start describing a little bit more, is that we have a very special climate. If we go back to that peninsula that I just showed, that mountain range really causes a lot of climatic influence with moisture. So these areas have high fog, they have high rainfall. And because these trees grow so long, canopy soils have the opportunity to form. And just to give you perspective in millimeters, because we're around the world and not stuck in a non-metric system, which I could talk about all day. But if I were standing there and all the rain in this forest were to fall at one time, I would be standing in 3,000 to 4,000 millimeters of water. So that is a lot of water. It's about 12 feet for my other US people who are sitting in here. I would be in over 12 feet of water. So a lot of water, not necessarily the wettest place on earth, but it does get notable mentions when they do make those lists of where's the wettest place on earth. And so what happens in these very wet, moist areas with these old growth trees is these thick mats of canopy soil form on these branches. They don't just form on the big leaf maples in this ecosystem. They form on other trees as well, but I focus on the big leaf maples because of their branching structure. It allows the mats to kind of get a bit more copious and stacked up. And so what happens is as these trees get older, the bryophytes, so mostly mosses over in this ecosystem, they establish on the tree. But then, of course, these trees grow over decades and centuries, and these mosses have their life cycles, which, they die, regrow, and they start stacking up organic matter. And then on top of that, we have other higher epiphytes, for example, licorice fern, other small plants. I've actually seen about a 5-foot sickest spruce growing 40 feet above ground in a maple tree. That's not a common occurrence, but it is an occurrence that I have seen. And so as all of these epiphytes establish, not only does their organic matter kind of stack up and break down, but it intercepts the other litter fall in these ecosystems. And what this does is it provides canopy soil. So underneath these living epiphytic mats, there is this canopy soil. And just to give you perspective, I mean, you can already see little me and huge tree, but the branch is about this large right here. So this whole thing it's kind of tricking you to think the branch is huge, but it's really the mat of canopy soil. And so what we really wanted to understand is canopy soils have really only been researched for about 40 years now. When I started, about 30 years. So it's a field that's pretty much in its infancy. One, because you have to get access to these pristine areas, which we don't want to harm in the sense of their nature. We want to be ethical about it. So permitting is really important. And then also just climbing trees, as I'm sure more of the people in this audience can relate to me than ever before, is actually pretty tedious in the sense of getting out there, getting structures up there, sampling and all of that. But what really sparked my interest is when we went out there and I started helping sample these canopy soils, I saw these roots under the soil mat. So this is a mat of canopy soil. I've seen canopy soil mats that are deeper than 35 centimeters. They can get pretty deep up there. So I saw these roots, and I was with one of my mentors at the time, and I was like, there's no large trees up here. Like, what are these roots? They're not, rhizomes or rhizoids from the bryophytes or the ferns. Like, these are large roots. And she said, oh, they're adventitious roots from the tree. And I was like, wait, what? So this tree is growing adventitious roots off of its branch into these organic soils. Organic soils are usually really tasty. They have a lot more nutrients than, for example, mineral soils. So I was like, whoa, there's roots growing out here. And that's when I was becoming, a young fungal enthusiast. So I was like, what do we know about the root-associated communities? What do we know about the plant available nutrients? Well, the answer to my question were really nothing. We don't know a lot. We know that there has been evidence that they're associating with fungi, but that's kind of where it stopped. And there's just so many things to still discover. And the one thing that really drove me into this was I was thinking about forest management, right? Climate change, old growth forest resiliency. And it kind of dawned on me that if we're ignoring the processes that are happening in these obviously extensive mats of canopy soil in this ecosystem, what are we missing when we look at these old growth forests and their survivability? Maybe the canopy soil compartment is something that has actually provided them kind of like this protective blanket in the face of a changing climate. So we wanted to look at the root associated fungal communities. We wanted to look at nitrogen and phosphorus dynamics. So how much nitrogen and phosphorus are available in the canopy compared to the forest floor. We also wanted to understand some carbon dynamics, for example, in greenhouse gas emissions and how that could plug into, for example, modeling scenarios in this system. We also more recently have looked at other biodiversity measures to overall just really understand how canopy soils influence ecosystem function. So I will run you through the results. I'm not going to go too much into methodology. If anybody is interested in the methodology, please feel free to ask me in an email. It can get pretty lengthy in time if we go into all of the methods. So what we wanted to do first was we wanted to look at fungal establishment in the canopy roots and the forest floor roots. So we collected 1,200 roots, we cleared them of their tannins, and then we stained the fungal organisms. These are using a mix of microscopy techniques, but overall, these are the fungal organisms established in the root. We can see that there's fungi in different layers of the cell. These all are overlapped with what you would see for mycorrhiza. So this is an arbuscular mycorrhizal associated tree, and these are arbuscular mycorrhiza structures. I had hypothesized that there wouldn't be as much fungal establishment in the canopy, and why this is important because it kind of gives you insights on the amount of exchange that may be going on in the plant or the amount of energy that is being put into maintain these relationships. For example, if there's not as much mycorrhiza in a certain type of rooting system, you can kind of get inferences to predict that they may not be benefiting that plant as much. But when we looked at a comparison between big leaf maple roots at the floor level and the canopy level, I thought that the canopy would have a lot less establishment, for example, because dispersal limitation, but we actually found that there was no significant difference, neither between the hoe and the queet site or between the canopy and the forest floor environment, which was amazing to me. So that's like, there's as much fungi in the canopy roots as there is in the forest floor roots. What kind of fungi are there? Do they shift under different climate change scenarios? And what does that mean for this host tree health? And so that's when we went into sequence and understand what fungal communities were associating not only with the old growth maple roots on the floor, because nobody had ever looked at that before, but also what was associating with the canopy roots. And so what this is a PCOA plot. It's supported by other statistical tests, but what it basically shows you is relationships in space. So this, for example, is an entire fungal community condensed into a distance point. So it's not just one fungal organism. This, if I look back at the data, would probably be about 300 different fungal organisms associating with one maple. And this is another maple, this is another maple, and so on. So these are all communities associating with the maples that I sampled. And what we can see is very beautiful in ordination space that there is a clear divide between the forest floor community and then also the canopy community. But then to add a different layer to that, there was also a pretty clear divide between the two different canopy ecosystems and the hoe and the queets, but not between the forest floor, again, which stacks up on that dispersal limitation. So we went a step further. And so after just comparing it at the floor level and at the canopy level, we went to the canopy level and I put in these drought structures because we are really interested in how these canopy soils may be threatened as these forests experience wetter winters and drier summers. So I actually set up structures 80 feet above the ground. It was a grand time, which diverted half the rainfall and then also doubled the amount of rainfall and then the ambient conditions. And what we wanted to know is, do we see different communities associating with these roots at different points in time? Because if we rewind, I mentioned that there can be fungal organisms that help trees with drought, for example. There can be fungal organisms that, can appear and be detrimental to the tree during certain seasons because of stress. We just really wanted to understand that. And so this one is not as clear as the last one. I was a little sad about that, but it's still supported statistically by a test called Permanova, which shows that they are significantly different, even though they're clustered a little more sparsely. But what we saw across these treatments was that in canopy soil environments, root-associated fungal community composition was not only dependent on the type of rainfall treatment, but it also changed on spatial location, and it shifted during the seasonal sampling period. So we were out there sampling these roots for an entire year in spring, summer, fall, and then spring again. Winter was too dangerous to climb that year, unfortunately. So through this study, we found a lot of species of interest. So for example, we found a group of fungi that's widely considered to be a decomposer, but there's been recent studies coming out that's showing that it actually can be mutualistic to a plant. And it was one of the most abundant species associating with our maple roots. So further functional studies are really needed to understand what it might be doing up there, because it might be doing something really significant. We don't know. We know it's there though. We found a lot of those dark septate endophytes, and some of them are dark septate endophytes that have been proven for other plants to benefit plants, for example, in times of drought, specifically this Cryptosporiopsis that we found a lot. Same thing for these other species. These are all facultative species that could be beneficial, that were up there in notable abundance. And overall, we had our Buscodimycorrhiza up there, which is an obligate mutualism. So there's really no question that there's a nutrient exchange going on between the plant roots and the fungal organisms. And so that was just a really cool takeaway, that we had a different diversity in the canopy and the forest floor. In the canopy, it was a little more prone to shifting over these functional seasons. So it's really kind of laying this foundation that fungal organisms and canopy soils may be pretty important in these old growth forests. We took it a step further to look at nitrogen and phosphorus availability. So we looked at mineralization, so the amount of nitrogen and phosphorus that's being made available in canopy and forest floors. The opposite would be immobile. So for example, if there's immobilization, there's no available nitrogen on the forest floor, there's really high competition. And so what we actually found was that canopy soils mineralize a lot more phosphorus and nitrogen than forest floor soils on a per mass basis. And that wasn't too much of a surprise, because these are organic soils and not mineral soils like on the forest floor. So the fact that they were organic means that they usually have a lot more organic matter that's breaking down and providing nutrients. But if we were holding a kilogram of canopy soil and a kilogram of forest floor soil, there is a lot more nutrients typically in the canopy soil available for those roots and fungi than there is in the forest floor. And there were some anomalies, especially in these later seasons. Fall 2018 and 2019 was when we wrapped this particular study. Of course, there's variability because it's a forest. There's always going to be variability. But what we found was that a lot of the times when there was no nitrogen or phosphorus available at the forest floor level, there was some available in the canopy level, which can be tapped into by these host trees. We did the same project for the drought and the rainfall. We also found that these structures and seasonality impacted these mineralization rates, which was not really a surprise, but it was staying consistent that these wetter and drier conditions do impact the amount of nutrients that are available at the canopy level. And the fact that sometimes there's not nutrients available on the forest floor, it becomes really important to think about when we think about managing these ecosystems and what canopy soils mean for the survivability of these trees. We converted this to a forest level basis. So I've been talking about a host tree basis. When we correct for canopy soils of big leaf maple at a hectare basis, we found that they are enhancing the annual nitrate pools by 5.2%, the ammonium pools by almost 50%, which is pretty significant. And overall, the net nitrogen by 8.7%, and then phosphate by 3.7%. And so those numbers, some of them are small, but the fact that at an ecosystem scale, they are enhancing the nutrient pools. And this is just big leaf maple. Sitka spruce harbors a lot more canopy soil and has a higher basal area in these stands. So if we were to incorporate spruce into this, those numbers I would hypothesize would just get a lot larger. So we're ignoring a lot when we don't think about canopy soils where they do prevail. They also, for example, when there's a heavy rainfall event, they leach these nutrients to the floor. Especially, they leach a lot of carbon, but these other nutrients that we looked at, for example, nitrates, ammonium, phosphates, those also leach to the floor. So even though canopies are kind of this separated ecosystem, they are looped in directly to these nutrient cycles in these forests. So then we look over to canopy soils being considered in the greenhouse gas cycle because we are just trying to build the story of not ignoring canopy soils because a lot of people try to, say they don't matter too much. This is a colleague and friend of mine, Lizzie Stone. She's amazing. She helped with this study and kind of led this end of the work. But we found that the canopy soils of just big leaf maple contribute about 4% to stand level CO2 emissions and contribute to about 4% of the methane sink. Methane is more potent than CO2. But the methane sink was dependent on soil depth. So the deeper these canopy soils get, the more they seem to kind of sequester that methane, which might do with the microbial processes. We don't know. We need more researchers to go out there and be canopy soil advocates. So overall, what we have found out is that there is a diverse, significantly functional microbial community clearly dwelling in the canopy soils, but these interactions we don't know too much about. Some of the work that is hot off the press, so you're some of the first people to hear about this, is that with the collaboration that I did recently with the team at Western Virginia University, we actually found that soil respiration was higher in the canopy soils than on the ground. Again, this makes sense. Respiration is decomposition into CO2, to put it very simply. But that makes sense because there is very active microorganisms up there and it's organic matter. So if you were looking at that, most people who work with soils and respiration would have hypothesized that. We also found that nematodes were more abundant in canopy soils, and the people who are working with this nematode diversity, I'm not a nematodologist, but the people who are were actually really excited. Apparently, they're finding some extremely rare nematodes up there, and they're finding more, for example, bacteriivores and fungivores. So nematodes that, for example, prey on the fungi and help the decomposition cycle, not necessarily pathogenic nematodes, which were also found, but a little more on the forest floor level. I looked at fungal root associates directly. This is the soils. So I mentioned earlier that there's rhizosphere, there's bulk soil, there's root. Fungal communities and bacterial communities can be different in all those levels. So this was looking at just canopy soils and not roots. And what we actually found was that bacterial and fungal diversity was highest on the ground and decreased with branch height, which we would expect because of those dispersal limitations. Not as many things can get into the canopy. But what we also found was that the soil communities were still more active in the canopy than they were on the ground. So there can be a lot of functional redundancy when there's a lot of diversity. So even though you have a high diversity, they may not be as functionally beneficial as a smaller diversity that has like a really tight niche of functionality. So canopy soils actually had more active beneficial communities than the forest floor collectively from all our research. The nematodes, microvores in particular, were more abundant in the canopy, which stacks on to these cycles that are going on up there. And the increased community activity and more trophic interactions, they're hypothesizing that this is happening because there's more interaction between nematodes, bacteria, and fungi in the canopy. And we also are seeing a lot more soil nitrogen in the canopy, which we're trying to link some of the drivers to that. So overall, canopy soils should not be overlooked or underlooked because they're above your head, because adventitious roots were associating with a unique suite of fungi, including mutualists, so they are getting nutrients from these canopy soils, which are acting as a nutrient reserve for the host tree, and not only the host tree, but the epiphytic diversity. A lot of the studies you'll see in canopy soil environments focus on the epiphytes. This one takes it to a different level of focusing on the host tree and then forest level function, which there we go. They contribute to forest level nutrient dynamics. And overall, I really have this, which is beginning to be a strong theory based on evidence, that they may be a resilience compartment for these old growth trees, especially in a changing climate. I do not see big leaf maples grow this old anywhere else. If you have, please let me know. But maybe I hypothesize that these maples can establish and live so long in these old growth forests because of the canopy soil compartment. There's a lot of things to still be sifted out, but overall, I don't have any doubt that canopy soils are important to these ecosystems. Everything I told you is just for big leaf maple. So just imagine what we would think if we start looking at Sitka spruce, which can harbor larger biomass of canopy soil. We found a wide array of mushroom diversity up there, and we're still teasing through that. Some of them are species that are not defined. There's so many people who have put time into this research. I could spend another, 40 minutes going over that. Also, thanks for bearing with me because I know I'm a little over. And that being said, this is my collaborator, Dr. Jen Kane. This is me, both of us in trees, where we are in our happy place. I'm happy to answer questions as we open up that part of the webinar.
That was awesome. Thank you so much. So interesting. Wow. Both of you fantastic. Thank you so much, Korena. Levon, would you like to come back, sir? Awesome. You'll see later on when we look through the chat, everybody was loving both of your talks. You can see them with all their little cartoon things that they like to press because they're children, basically. I have put in the chat just now, but we'll only have time to work through a few questions, but that's fine. We'll send them all to our speakers afterwards. So we will jump around for a little bit, and we will go back to Levon for a minute. Peter's asked about the fact that in the UK, urban trees have an average lifespan of about 20 years. We sometimes hear it's about 13 years, but up to about 20 years. Is this a similar situation in Philadelphia that you found?
So we didn't